Hi, so my name is Sandro Santagata. I'm a pathologist at Brigham and Women's Hospital. I'll tell you today about some of the work we're doing in multiplex tissue imaging and challenges and opportunities. Um, I don't have financial disclosures, but I will talk about a project that we have with RareSight through an SBIR mechanism. Um, so, so my objectives for this talk are to give you a sense of the approaches we use for antibody tissue imaging, sort of the practical view of the tools and technologies, as well as the software for analysis, visualization, and sharing. I'll talk about three main approaches: Cyclic, which is our research tool; Orion whole slide imaging, which is intended for clinical translation; and 3D cyclic immunofluorescence, which is more of a cell biology discovery approach. And the applications, as you can imagine, for all these technologies are pretty vast on normal and diseased tissue, both in FFPE with preserved morphology, but also on frozen when integration with techniques like mass spectrometry imaging are necessary. And again, the applications span basic biology, tissue biology, and clinical work, as well as atlasing. The work we do is done in the Laboratory of Systems Pharmacology at Harvard Medical School. Peter Sorger is the director of the LSP. Laura Malasevsky is the executive director. It's a multi-institutional space. Lots of collaboration with a lot of talented people across software engineering, pathology, oncology, et cetera. So it's a really wonderful, dynamic environment. All of the work we do is posted to our website, tissueatlas.org. I'll highlight the websites that I think are important in yellow, and thanks to our funding agency, Ludwig, Gray, and the NCI. I'm a pathologist, so we spend a lot of time diagnosing disease. We have pretty impressive tools thus far, enabled further by genomics, but there's still a lot of information that's left within tissues. So we're really driven to extract more and more information to improve classification, risk assessment, the interception of disease, as well as treatment and outcomes. But the main driver for this is the need for improved discovery, and that's one of the goals of the Human Tumor Atlas Network, which we're a part of, the H10. And this is our perspective article written here, which I recommend you read if you want to have a view of the goals of tissue atlasing for improving human disease. The good part about the spatial omics field is that it's going to be based upon 100 years of histopathology by tens of thousands or more experimental and practicing pathologists. So we know the features that are critical from a morphology perspective. And now what's important is to be able to layer on key molecular information. I'll focus on the antibody-based methods, but we have work across with the RNA methods and and mass spectrometry imaging as well. The principal research method that we use is called cyclic immunofluorescence, CyCIF. It's one of a family of fluorescence microscopy methods. I'll run these videos on the side. It's pretty straightforward. We use several antibodies, 3, 4, 5, conjugated to different fluorophores, applied them to an FFPE section, the ones we use in pathology practice, add a DNA dye, image, photo bleach, and do multiple cycles of the imaging. And then we stitch and register the images and go off into computational space. The reason we like this method is that it's really simple. It's very affordable, about $100 to $200 per slide in terms of reagent costs. Everything's in the public domain, available. The reagents are widely available. You can change reagents mid-experiment if you see something you want to focus on further. We have methods for reagent validation, which I'll present in the next slide. It's easy to perform. We have anywhere 15 to 20 people in the group at the LSP doing imaging at one time, and it provides us with whole slide imaging. The challenge with whole slide imaging is that there's a lot of information, so we've had to develop a whole suite of tools, some of which Robert Kruger presented earlier, which is great. Congratulations, Robert, for his new position at NYU. But these tools are pretty remarkable in terms of how much work went into them, and I'll talk about them in a second. In terms of antibody validation, do antibodies work? Traditionally, we have a whole set of methods that we use in the lab to look at the quality of antibody staining in IHC and IF. There are a whole set of well-established methods, but with multiplex imaging, you can actually do even more. You can quantify, for instance, 5 different antibodies to a single target. Here are plots of pixel-level correlation between different antibodies to PD-L1. So you can see that this one clone, E1L3N, is highly correlated with Cal10, but other clones are not highly correlated. So you can start to use methods like this for quantitative antibody reagent validation for difficult to study molecules, signaling molecules, post-translation modifications, et cetera. We present the approach in this Nature Protocols paper and recently published an approach in breast cancer as well. You get these slides, they're rather large, they're composed of many different tiles, often hundreds of tiles that have a tremendous amount of informative subcellular resolution data that need to be stitched together. Each tile has multiple channels, and then again, you need to stitch them across and then do registration across cycles. So rather challenging. So the group developed a software by Jeremy Mullich called Ashlar, named after the Ashlar masonry. You can find that at our GitHub and also in this manuscript here. Moreover, when you want to dig into your data sets, it's critical to have very well annotated metadata. We've published a minimum information guidelines, MITI, for high-plex tissue, both for clinical metadata, biospecimen metadata, and experimental metadata, basically what to report in manuscripts and to keep internal records. The pipeline that Robert mentioned is called MC Micro, so it's a scalable, flexible Nextflow language pipeline with different modules that you can swap in and out. So it's very customizable. It takes you from the images all the way to a raw counts table, a spatial features table. There's a graphical user interface as well as command line for those that choose to do so. In addition, there's the need for education and outreach. And again, we recommend that you visit us at tissueatlas.org for that information. One of the additional challenges with whole slide images is that they can have artifacts, their tissue folds, their antibody aggregates. There's loss of tissue that can come with cycling. So Greg Baker and the group developed Silentor. It's an interactive quality control tool for reviewing in this dashboard setting the various types of artifacts that can occur. So a human in the loop review, also using data analysis. Certain clusters may be erroneous and artifactual, perhaps at the edge of a slide. So that can be the raw feature table that comes out of MC Micro can be analyzed through Silentor. You get the redacted feature table and then off into computational analysis. And you can find Silentor again at our GitHub. And then the other challenge with big data is kind of big data, big problems is how to share it. So typically in a manuscript, you have a figure like this, and you have this tiny little postage stamp of the data. And that's just an injustice to the remarkable data that all the members of this community are generating. So inspired by the type of pathology we do review, where someone sits at the microscope and educates the trainees, and also inspired by museum tours, narrated museum tours, both online and in person. We developed a tool called Minerva, which is, it permits people to write their own stories. You have a table of contents. People can be marched through, walk through a tour of the specific images. And then the viewer can also disengage if they want, go on, sort of ramble through the museum on their own and look for the features that they're most interested in. So this is available. This tool is called Minerva. We described this in a manuscript a couple of years ago in Nature Biomedical Engineering. There's an authoring tool. There's also a Minerva story, which is the actual story itself. And now we're embedding Minerva into cBioPortal with Nikki Schultz and Eno De Bruyne. So what are some of the applications for methods like SciSif? They're vast, so there are many projects ongoing at the lab at one time. I'll just focus on a couple. In cancer, obviously, cell proliferation is critical to measure. We typically do this in the clinical lab using one marker, a single marker, Ki67, which is fraught with issues. You're not sure if you're looking necessarily at a tumor cell. So that might be a lymphocyte. There's variable staining very low. Is that a positive cell? Is that a positive cell? And the reason is that Ki67 has a graded expression that comes up in the G2 phase of the cell cycle. So to start to get around some of these flaws with multiplex imaging, you now can use multiple markers. We can include markers that tell us if a cell is arrested and not cycling. We can include multiple markers. Ki67 is a marker of proliferation, but so is PCNA and MCM2. So we can find cells that are Ki67 low or negative and support that they're proliferative using additional markers. Moreover, we can use lineage markers to identify whether it's a cell is a tumor cell or an immune cell, and then focus our analysis exclusively on the tumor cell populations, separating them into arrested, non-proliferative, or proliferative cells. And then we can spatially map these back onto the tissue. This is a breast cancer case where you can see in blue, these are the quiescent areas, and green are proliferative areas, and red are arrested. So you can see there's a geography to proliferation in cancer tissues that is somewhat surprising to see. And in collaboration with Judith Agudo and others in the Boston area, we're now trying to understand the features and the factors that drive these proliferative clusters of cells and their role in therapy resistance. Moreover, we can then isolate those proliferative cells computationally and then look at other markers that are being expressed, for instance, cell cycle markers that cover G1, S, and G2. And the distribution of these markers can tell us whether the population of cells are skewed in one manner, if they're evenly distributed, the way they would be in a typical cell culture plate, or if there are factors that are driving G1 or G2 skews, or even arrest. So there's a lot that can be done with these types of metrics. And moreover, part of our goal is to develop increasingly better metrics for measuring tumors and understanding their properties. So in another manuscript, instead of describing just the quantity of immune cells, we're trying to describe actually if they're in networks. So what we see is that you have these large tertiary lymphoid structures, which are linked to good patient outcome in many cancers, but there are many smaller lymphoid structures, often on the size of 10, 20, 50 cells. And now we can quantitatively describe these with graphic methods to assess what are the constituents, what are their properties, and what do they mean for cancer therapy response. Additional work that we've done using cyclic immunofluorescence is to take serial sections through, this is like a first generation 3D atlas where we took serial sections through a colorectal cancer specimen. One section we would do an H&E, the neighboring section we would do cyclic immunofluorescence, and we did this 25 steps through a specimen and then made a 3D reconstruction. I won't go into all the details of the study, which was published last year, but there is a remarkable connection between the molecular features and the histologic features, and there are a whole wide range of transitions that we see. When we look at this three-dimensional reconstruction, it's pretty amazing because the 2D features, which, for instance, these individual budding tumor cells into the stroma, when we look at them in 3 dimensions, they're actually not individual cells, but they're actually these long fibrils of interconnected tendrils basically sticking out of the tumor mass, projecting into the stroma, and we see molecular gradients of EMT and other features along those tendrils. If we look at the mucin pools that are present throughout the tumor, and then we do a 3D reconstruction, we notice that they're not pools, but they're actually interconnected caverns. This kind of like this kind of cavernous structure where you can actually enter the portal at the lumen of the colon and then follow your way all the way to the invasive front. So you see these impressive structural features of cancer. In addition, tertiary lymphoid structures, which are present throughout the invasive margin, even within the tumor, we can annotate these histologically and molecularly inside CIF, and then find that they're actually not individual lymphoid structures, but actually lymphoid networks. They're broadly connected across the tumor stromal interface. So there's a lot of really interesting information. Moreover, what we see is if we look at the molecular features, there's a lot of gradation of molecular features across specimens. So here, if you look at P53 or EGFR, of the 100 markers that we have, there many are graded or heterogeneous in this fashion. You can see that there's a large amount of variability in expression. And what's important here is, well, conceptually, it's really interesting. What's driving this molecular heterogeneity and these gradients? Are these morphological kind of gradients akin to what we see in development? Are they subclonal populations? There's a lot of information still to gather. But if you look at these circles, these are the cores from tissue microarrays, and you can get a sense already of the subsampling that occurs when one uses tissue microarrays. So we think is that there are very important considerations when doing tissue microarray studies in terms of adequate sampling and the statistics that one needs to use to derive conclusions, particularly when trying to make links to patient outcome. Because due to the central limit theorem, a lot of the cells that are present in a single core are 1,000, 2,000 cells. They're spatially correlated in their phenotypes as well. So in effect, if you're measuring 2,000 cells, you might only be measuring 1/100th truly in terms of independent statistical events. So these are important features for antibody imaging, spatial transcriptomics, and also when you consider biopsies in a clinical setting as well. So one additional challenge, though, multiple challenges you have with cyclic methods is that many samples do really well with cycling and some don't. Some have small scale deformations that can lead to problems, hence the need for silencer. Some samples, particularly core biopsies, or let's say breast, fibrous tissues, for instance, can degrade with cycling. There can be imaging artifacts that occur in terms of the DNA stain, and the quality of the H&E at the end is not quite what you'd like for a clinical grade H&E. So inspired by this, we've worked with Raresight, which is a company in Seattle that does amazing microscopy through an SBIR grant on the Orion method. And to do that, we sort of stepped back and thought about the first principles we'd like in a clinical system. We wanted whole slide imaging for the reasons I mentioned before, a reasonable amount of plex that pathologists could review and reasonably report. Same slide immunofluorescence and H&E. We're surprised by the quality of the H&E after this because there's no cycling. Subcellular resolution is important to help with segmentation, and then simplicity. You just want a one-shot method that can actually operate same day in a clinical lab in a manner that's expeditious and reliable. So Raricyte developed a method in which we can apply 16 to 18 antibodies, each conjugated to a different fluorophore onto an FFPE tissue using fancy microscopy methods that I won't go into. We can now separate those different signals. We take the same slide, apply it to a stain, apply H&E staining, and then put on an automated stainer, and then we can merge the images, as you can see here in this lensing tool developed by Robert Kruger. You can now slide around and look at the H&E, but also look at the IF from the same tissues. So we reported this in a manuscript last year at the lower left. You can view the images of samples in our GitHub as well. And now you can bridge morphology. This supports histopathology workflows, figuring out the molecular properties of certain cell types, and also allows for integrated machine learning, as well as a two-way transfer of information in studies where you have information in the H&E world and you want to bring it to the immunofluorescence world, and vice versa. I'll be pretty brief in terms of the applications. There are many for this, and in our paper we describe a few of them. In colorectal cancer, we recapitulated a biomarker score called the immunoscore, which is based upon immunohistochemistry for CD3 and CD8, but instead we use our immunofluorescence and our other markers to guide our measurement of the levels of the immune infiltrates, both at the tumor edge, and it's also scored. Those infiltrates are also scored within the tumor itself. Using the immunofluorescence data, each tumor is given a score. High is associated with high infiltration and a good outcome. Low score for infiltration is associated with a bad outcome. And we can recapitulate this with using the Orion method in a very quick fashion. Moreover, because we have more markers, we can take the same logic of looking at the expression of immune markers and other markers at the tumor stromal interface and within the tumor. And then we can compare how effective they are, their hazard ratios at predicting good and bad outcome tumors, and we can rank them. And the immunoscore, our version of it, which we call Image Feature Model 1, does very well. But there are many, even hundreds, that are combinations of markers that are better. So you can use this as a discovery platform for finding the optimal biomarkers and then testing those in validation cohorts. What I want to do in the last few minutes is tell you what I think I'm most excited about, even though I love the clinical part as well, is the cell biology type discovery work that I think we'll be able to do now with a new application, which is using cyclic immunofluorescence in 3D. So if we take a standard 5-micron section and we then do Z-stack imaging through the specimen, you can see that almost every nucleus is chopped in half. You're only getting a partial view of any cell. If you want to measure the volume of a cell, you need to have the whole view of the cell. So with thicker sections, not too much thicker, 20 microns, 30 or 40, whatever can actually physically sit on a slide, we can now do cyclic immunofluorescence on thicker sections and get full profiles of nuclei and full profiles of cells. Combining this with additional methods for non-labeled imaging of collagen, second harmonic generation, we can get high-plex multiplexing and additional extracellular matrix on samples such as this melanoma in situ specimen. And what's pretty remarkable at these higher resolutions, you can quite remarkably see single cell features. These are CD8 cells. They're forming little communities, the lymphocyte networks that I mentioned before, that of cells that are lymphocytes that are forming these little communities. Here's a B cell that's crawling along the collagen fiber with interactions with CD4 cells. And if I just turn this video on, you can see that there are complex interactions with the cell membranes of CD4 cells and CD8s that are harder to appreciate in standard 2D imaging. And even cell typing is remarkably enabled, I would say, by this high resolution imaging that's now possible with sort of these newer methods of cyclic immunofluorescence and confocal microscopy. And here I just want to end with this kind of view of the future, I think, where we're going to hopefully be able to link the imaging we do in cell lines with the imaging we do in tissues. So here's a glioblastoma cell that's about to have its primary nucleus rupture. That's a marker, BAF, of primary nuclear rupture that flashed over there. In the next video, you can see cells dividing, a micronucleus forms. The cell's going to divide as well. Another micronucleus is going to form. And when those micronuclei rupture, they leak their DNA into the cytoplasm. And again, the same molecule, BAF, RET turns on at the micronucleus and will turn on at this micronucleus. And then in this final video, you see a cell here that's about to divide. Um, when it divides, the two daughter cells are linked by a chromosomal bridge over here. And again, that's highlighted by that same BAF protein. We can see these in standard 2D imaging with primary nuclear ruptures in glioblastoma. There's the actual nuclei rupturing and then repairing, micronuclei that also rupture and undergo damage. With higher resolution imaging, we get great views of those micronuclei. This is a ruptured micronucleus with C gas and sting, and I'm not showing you here, but also gamma H2AX, so it's undergoing DNA damage, the substrate for chromothripsis. With the fixed section 3D, we can get these beautiful views of chromosomal bridges between cells, which are pretty remarkable. We can also render these in 3 dimensions using a variety of tools to measure the shapes of the cells and the properties of those cells that have chromosomal bridges. Here's another bridge of going from one primary nucleus to a micronucleus of another cell. And then just to give you a sense of like what we're starting to see, this is the Z plane here. So you can see that we get whole profiles of nuclei. This nucleus has a rupture in its primary nucleus. This cell here has a micronucleus that's connected by a bridge to this other nucleus. And if you were only taking 5-micron thick sections, you would only get a very impartial view of what's occurring in these samples. And this is just, I guess, the broader future where we're doing deep 3D SciSif of millimeter-sized pieces after clearing and using light sheet microscopy. And you can get, again, a view of the nerve fibers coming up, the keratine-positive epithelium of this colon here, of lymphocytes, and starting to get the larger architectural arrangements that are possible with this type of imaging. So in conclusion, there are powerful new tools that are available to us, 2D methods for multiplexing both on the research side and the clinical side. We're trying to move some of these assays into the clinic, hopefully in the not too distant future. 3D methods are also very powerful. The software tools are developed, but still in need of more development, and there's a lot to be done in that world, and we're excited about that. And then it's about selecting the right approach. It really depends on your question. And we're in an important phase. We're definitely in an important phase of description of tissue features, applying these from cell lines to mice to humans in clinical trials, and bridging the world of computation with pathology, which I think is really key for the future. And we're finding, I think, interesting features. I want to thank the team, particularly Peter, who I do all this work hand in hand with, Laura Malsevsky, our executive director, our program managers, and our collaborators and wonderful scientists at the LSP. So thank you very much.
Thank you.